Hey, Hammy here, back with part 2 of section 6.1, where we're going to look at Mendel's first set of crosses and what he thought was going on there. In some of the very first experiments he did, he took his pea plants and he let them self-pollinate for many generations until he got what we call pure lines. Okay, sometimes in like dogs, we would call this purebreds. In other words, pure lines, he knew that if he let them self-pollinate, if they were tall, they would always be tall. If they were short, all the offspring would be short. He knew that they would always give rise to that same characteristic. Well, he started with flower color, with pure purple and pure white, and he crossed them. So this is referred to as the P generation, P standing for parent, okay, the parental generation. So he crossed true breeding types, and when he crossed pure purple, okay, pure purple over here, with pure white over here, okay, he created what we call a hybrid, okay, a hybrid between the two pure lines. This offspring of these two are called the F1. Okay, F stands for filial. So F1, first filial, comes from the Latin and Greek for sons and daughters. So it's you have the parents, now you have the first sons or daughters. And something quite astonishing happened. When he crossed purple flower and white flower, look at the F1 generation down here. Do you see any white flowers? Don't think so. He was astounded that the white trait disappeared. Where did it go? Okay. And he thought, well, that is really strange because it's not blending theory. They didn't turn kind of a whatever white and purple mixed together make, but they were all purple, like the one parent. So next, he allowed the F1 generation to self-pollinate. Okay, so he took the F1 generation here and let them self-pollinate or cross-pollinate, because they were all purple flowers, and that produced what we call the F2 or second filial generation. And voila, what do you think he noticed? Yeah, that white trait reappeared in the next generation. Okay, and he noticed when he did these crosses, it was always, okay, again, the biology, doing biology experiments, but the math brain, being good with numbers. He picked up on a pattern. He noticed for every 3 purple flowered plants, there was 1 white flowered plants. It was always this 3-to-1 ratio. And when that white trait kind of reappeared in that second or F2 generation. So Mendel began experimenting with the other traits. He actually went through all 7 characteristics that we had mentioned earlier in the previous video, that the reason that he was using pea plants for these crosses. And in each case, one of the characteristics disappeared in the F1 generation and then reappeared in the second generation, and it was always approximately a 75% to 25% ratio, okay, or a 3-to-1 ratio. So there was always 3 times as many round as there were wrinkled seeds. Same thing with yellow to green, white flowers to purple, which actually purple was dominant. So purple to white, full, constricted. And it was in all of these crosses, one of these traits, okay, either top or bottom for each of the characteristics here, 1, 2, 3, 4, 5, 6, 7 different traits. He noticed that it was always a 3-to-1 ratio when it reappeared in the F2 generation. This led him to one of his first principles, the law of segregation. Okay, now when we think of segregation, as that was a really bad thing in the South in the early in U.S. history. So segregation means to separate or keep separated. Separation in this case, when we talk about genetics, he stated that there were 2 factors controlling a given trait, okay, one which dominates the other. So we talk about dominant versus recessive. So the purple trait must have been dominant to the white trait, okay? And they had 2 Factors, we now know that these are genes. They had two genes, or factors or alleles controlling that trait. And when they produce gametes, the ovules and the pollen, okay, which would be sperm and eggs in animals, but in plants, it's pollen and ovules. When the plant reproduces, okay, we now know that it goes through meiosis. Okay, and remember in meiosis, we were concerned about what was happening with those chromosomes in the nucleus. Okay, he noticed that the sister chromatids in meiosis 1, we pulled homologous chromosomes apart, and then in the second part, we pulled the sister chromatids apart, so that in the gametes, you had each of the single sister chromatids, or that copy of that gene or allele, was pulled apart from all the other copies, and now you had an equal chance to recombine with the other parent to make all the different combinations. And this allowed him to come up with some of the characteristics and the ratios that he did.